Hey all, I am A. Vignesh Shankar, pursuing second year biomedical engineer, SNS College of Technology, Coimbatore. So today I am going to talk about my article symbol staining. Symbol staining is one of the type of symbol procedure that can be used in symbol stain, in which only one stain is used. All types of bacteria appear as a color of the stain when viewed under the microscope. Some stain commonly used for symbol stain include crystal violet, saffron, and methylene blue. Symbol stains can be used to determine the bacterial species, morphology, and arrangements, but they do not give any additional information. Living bacteria are almost colorless and do not present sufficient contrast with the water in which they are suspended to be clearly visible. The purpose of staining is to increase the contrast between the organism and the background so that they are mostly readily seen in the light microscope. In a simple stain, a bacterial smear is stained with a solution of a single dye that stains all centers the same order without differentiation of cells, types of or structures. The single dye used here in our lab is methylene blue, a basic stain uh, as a basic stain. Okay, the basic stains having a positive charge bind strongly to negatively charged cell components such as bacterial nucleic acids and cell wall as well. That's all about my topic. Thank you.
In this video, I am going to talk about gram staining. Gram staining is a common technique used to differentiate two large groups of bacteria based on their different cell wall consistence. The gram stain procedure distinguishes between the gram positive and gram negative groups by coloring these cells red or violet. Gram positive bacteria stain violet due to the presence of a thick layer of peptidoglycan in their cell walls, which retains the crystal violet. These cells are stained with. Alternatively, gram-negative bacteria stain red, which is attributed to a thinner peptidoglycan wall, which does not retain and crystal violet during the decoloring process. Gram staining involves three processes: staining with a water-soluble dye called crystal violet, decolorization, and counter-staining, usually with the safranin. Due to differences in the thickness of the peptidoglycan layer in the cell membrane between gram-positive and gram-negative bacteria, gram-positive bacteria retain crystal violet stain during the decolorization process, while gram-negative bacteria lost the crystal violet stain and are instead stained by the safranin in the final staining processes.
Hello everyone, now I'm going to explain 3 steps on gram staining. Cells are stained with crystal violet dye. Next, a gram iodine solution is added to form a complex between the crystal violet and iodine. This complex is a larger molecule than the original crystal violet stain and iodine and is insoluble in water. Decolorization such as ethyl alcohol or acetone is added to the sample, which dehydrates the peptidoglycan layer that strings and tightening it able to pregnate this tightened polyglycogen layer, and this is trapped in a cell in gram-positive bacteria. Gram-negative bacteria is degenerated and the thinner polyglycan layer of gram-negative cells is unable to retain the crystal violet iodine complex and the color is lost. The third step was the crystalline stain such as weakly water soluble saffron is added to the sample. Staining is red since the saffron is tightened than crystal violet. It does not disrupt the purple colorization in the gram positive cells. However, the decolorization gram negative cells are stained red. Thank you all.